Gopalakrishnan, who is an assistant professor at the Center for Translational Medicine, University of Maryland School of Pharmacy. Dr. Gopalakrishnan is a quantitative clinical pharmacologist and a biostatistician who has over 10 years experience using advanced quantitative approaches to help with developing different kind of models. She uses these advanced, innovative, quantitative approaches to guide precision therapeutics in maternal, pediatrics, and clinically ill patients. She has over 50, she's authored over 50 peer-reviewed publications. She's a recipient of American College of Clinical Pharmacy Best Teacher Award in 2018. She's also the director for the online master's program in pharmaceutics at UMB. Today, she's presenting on precision therapeutics guided by pharmacometrics in lactating women. Please take it away.
Thank you very much for memorizing my bio and saying it. I really appreciate it. Good morning, everybody. It's my pleasure to be here, and thank you to Indiana CTSI, Dr. Sarah Quinney, for the invitation and the opportunity to present our research in this area, which I've been recently associated with both in pregnant and lactating women, and I would like to share our research here. I know I'm between you and the lunch, but we will get there, don't worry. So prescription medication use is common in pregnant and lactating women, as it is with any other population. Dr. Fletcher actually in the morning set up the stage for this. People working in the maternal research area are familiar with the facts that 60% of pregnant women take at least one prescription medication. 70% of lactating women, breastfeeding women take at least some form of prescription medications, which is common for treating chronic illnesses. However, the point is majority of these medications are used off-label. So that is where the reason behind this is because of gaps in knowledge, because the studies have not been done in these special populations to inform labeling. So one study reported that there is a delay of 6 years for the label to be updated to include some kind of dosing information for pregnant or lactating women. And the reason is, as you can see, among all the trials, only 0.3% of active registered trials are pregnancy drug trials. And among them, if you take it, less than 5% have some kind of PK evaluations that can potentially inform labeling about dosing for prescription medications in pregnant and lactating women. So there is a big knowledge gap here. So in preparation of this talk, I just wanted to go a little bit back in the history to understand why this is the case and what has been done in the last few decades. So it all started with the thalidomide tragedy, and it was taken off the market by FDA in the 1960s. And in 1970s, because then FDA had more, increased regulations of evaluating the drugs. So the FDA guideline in 70s, which is the general consideration for clinical drug evaluation, prevented pregnant women in participating in trials. And subsequently in 1979, they had a pregnancy category labeling system, which labeled pregnant women as vulnerable population. So that kind of restricted, including pregnant and lactating women in participating in clinical trials. So however, since then, there has been several initiatives by NIH and FDA to increase inclusion of pregnant and lactating women in trials, starting with the National Institute of Health Revitalization Act, which necessitated required inclusion of women in clinical trials and research But the last two decades, PK trials in quite a few drugs. Then came in the pregnancy and lactation labeling rule, PLLR, in 2015, which FDA required the sponsors to include any kind of information that's available regarding use of prescription drugs in pregnant and lactating women. Majority of the labels did include animal studies where they did not mandate human information, but that came into effect. That kind of put some pressure on the sponsors. And there were guidances by FDA about the considerations of conducting PK studies in pregnant women, and also ethical consideration, a recent guideline about ethical consideration of inclusion in clinical trials for pregnant women. And at the end of the few years back, because of the 21st Century Cures Act, we have the PregLac task force established, which has come up with 15 recommendations that was submitted to HHS in 2020, and that has consequences, and hopefully it is an encouraging improvement in this area. And one important thing I also wanted to point out, with Dr. Rehman mentioned in her presentation yesterday, was also in 2019, the modified common rule removed pregnant as a vulnerable population. So you can see there has been progress made, but there is still a lot more to do. So I kind of looked at to see that there were several ongoing trials in pregnant and lactating women. Some of the important things that I wanted to point out as part of Pediatric Trials Network was the CUDDLE study, where they studied PK and safety of commonly used drugs in pregnant women, in lactating women and breastfed infants using opportunistic PK sampling. There is a wide variety of drugs across different therapeutic areas that are being studied in this trial. There is a recent trial which started in January 2024 about anti-HIV medications in breastfeeding women in Botswana. There is a good amount of studies that have been done in the HIV space to inform labeling, which is through the IMPACT network, which probably some of you are very familiar with. So I also did a crude survey to see what are over the past 10 years, how the trials registered at clinicaltrials.gov in terms of PK evaluation. So certainly you can see there is a consistent progress, and especially in the past 2 years, there were about 15 trials which included studies in pregnant and lactating women. So it's moving in the right direction, if you want to say, and it's an encouraging direction, and that provides an opportunity for using precision therapeutics and using pharmacometric principles. So having said that, I would like to highlight 2 case studies as where in our research where we had information or where we studied information in lactating women, and we leveraged pharmacometric models to inform our precision therapeutics. The first project, though I talked about prescription medication, the project that I'm presenting here is on low-calorie sweeteners. These are food additives approved by FDA, whatever is applicable in prescription drugs. This is something which we commonly use. As many of you might have taken, Splenda, it's also one of the low-calorie sweeteners. Equal, which was there outside, that all has, it's a low-calorie sweetener, which is the one I will be talking about today. And this is a project that came to us in collaboration with Dr. Alison Silvetsky from George Washington. Brooke, my graduate student, she's working on this research. The other case study, following Cindy's talk, she talked about the non-psychoactive part of cannabis. I'll be talking about the psychoactive part of cannabis, which is the THC. This is my another graduate student, Jide, who's working on cannabis, studying infant exposure via breast milk in recreational cannabis users. So let me walk you through these 2 case studies to demonstrate how we can leverage these quantitative approaches in this less studied area. So With respect to the low-calorie sweeteners, sucralose and ACE-K are the two commonly used low-calorie sweeteners. ACE-K refers to acesulfame potassium, and you can see a wide variety of food products, beverages containing these two low-calorie sweeteners. You may be surprised that not only the diet products, but also other products like the yogurt, the general Gatorade, fruit cups, whole grain products here, Pedialyte. You can see the two low-calorie sweeteners I mentioned, pretty much widely used. And I mean, after being involved in this project, I've become a careful reader of all the labels to make sure what we are taking in. So this really has opened up quite a lot of things in terms of like day-to-day observations. So you can see how commonly these are used. These are FDA-approved food additives. But the point why we need to be studying these is for the past like 10 years or so, there has been an inundation of articles by WHO, by New York Times, by Harvard Health, talking about the health implication of these artificial sweeteners. There has been quite a few studies where there is substantial evidence that has been collected that these use of artificial sweeteners are associated with increased risk of obesity, metabolic diseases, type 2 diabetes, along with hypertension, and also the change in the microbiota, and also leading to the gut microbiome changes. And also several studies have shown that if an infant is exposed, early exposure in infants could lead to their changing their taste preferences. They may be starting to prefer more sweetness. So which could potentially have implications later on in their life about metabolic disorders. So there is an need to at least understand what is the levels that we see in infants. And this is one statistic about lactating women in particular, that it is in a survey which said 44% of lactating women consume LCS containing beverages and food products. So there is a lot of thing riding on this. And so the question was, what could be the health implication on mother and infant, and potentially in terms of what we know about in terms of a breastfeeding infant. So that led to our question, which is, so what should be the recommended LCS consumption during lactation? Any food product containing low-calorie sweeteners. Specifically, we are looking at sucralose and ACE-K because those are the more predominantly found sweeteners, so that could potentially inform breastfeeding guidelines. So I put it in a chart like something called as a DIA. Coming from yoga school, we have been taught to think in terms of what is the key question, what is the key decision that we need to make, and then what information we know, and then decide on what analysis method we need to use. So here, the information we had was, it was a prospective controlled single dose PK study of sucralose and ACE-K in lactating women and their infants. So the women consumed ocean spray diet cranberry juice, 20 ounces of them, which contained both ACE-K and sucralose in the doses that's mentioned. So this was the data that we had, and we utilized population PK modeling approaches to understand or at least to predict upon repeated administration, what could be the potential level that we would see in an infant, in a breastfeeding infant, and what are the subsequent clinical implications of that. So let me walk you through the study design. So this was a prospective study. It was single-site interventional study. It was done in George Washington University. The study beverage was ocean spray diet. The study population was 40 mother-infant diets, which and they were 6 months, less than 6 months postpartum, and they were exclusively breastfeeding. And the infants ranged in ages between 4 weeks and 6 months of age. And the rich sampling was collected in maternal plasma and breast milk, and 1 infant heel stick was obtained, along with other baseline covariates. And the mothers maintained food diaries 1 week prior to the study. It was so we were able to make sure that they did not consume any food with LCS. That was ensured. So this is the data that from the plasma and breast milk levels for the two LCS, the sucralose and ASK. This is sucralose. The darker on the y-axis is the concentration, the x-axis is the time. And you can see the plasma, and this is the breast milk levels. Definitely here for sucralose, the breast milk levels are much lower than what you see in plasma. So the Cmax and AUC were 7 to 9-fold lower in breast milk. And in infants, out of the 40 infants, we had about 15 of those infants who had measurable levels of sucralose. And the mean infant plasma concentration was 5 nanogram per ml. And this is ASK, the other sweetener that I was referring to. Here, it was a different story. The breast milk levels were at least 2-fold higher than plasma. And all infants showed 40, like all of the 40 infants had measurable levels, and the mean infant plasma concentration was about 9 nanogram per ml. So the data pretty much gives us an idea about what is the level that we are seeing in infants, but this is from a single administration. And we had to, also we have to understand what are those clinical implication of the levels that we may find upon repeated consumption. This is, I mean, in regular day-to-day life, a mother can consume multiple times the beverages or any product containing the LCS. So we used a POP-PK approach because we had a good amount of data in maternal plasma and breast milk. So what you're seeing here is a 2-compartment model for maternal plasma to describe the maternal plasma levels. And we used as you saw in the previous slide, there was a delay in the appearance of levels in the breast milk. So we kind of used a delayed absorption compartment or delayed transfer compartment into the breast milk by having a transit compartment here. And we tried out several different approaches, and we settled on something estimating a partition coefficient of plasma to breast milk ratio in explaining the breast milk levels. So I will be showing the results from the sucralose modeling, as the other compound is we are still working on it. So now we have this maternal information for the infant. The main question that we had was the dose determination. How do we account for the infant dose? Since these mothers were exclusively breastfeeding, initially we did not have, I mean, it was hard to know how much exactly the volume consumed by the infants. And there were some methods that we used or that they used during the trial to weigh the baby before and after the breast milk consumption to understand what was the change. There were several approaches we tried, and some others did pump. So there was a lot of variability in that. So we decided that we will use Cindy's equation to determine weight normalized human milk intake. And we assumed 8 feeds per day, which is the typical average feed volume to calculate the infant dose, and then scale these maternal plasma parameters to infants to predict in infants, which we are still working on it. So the idea is these two compounds, the sucralose and ASK, have different physiological characteristics. Sucralose is less absorbed, it's completely excreted unchanged. So all those things have to be kept in mind. And when we adjust for in infants, those parameters, those things have to be taken into consideration. And there is no metabolism anywhere, so there was no age maturation that we need to consider when we scale down to infants. So these are the results from the population PK analysis. These are some representative profiles. The black dots indicate the observed concentration. The green lines are the model predicted. This is for plasma in sucralose, and this is the overall fit. As you can see, the model is able to adequately describe the concentration. We had, there was at least about 50 to 60% variability in clearance and volume parameters. There were no specific covariates that could explain the variability between subjects for plasma parameters. This is how it looks in breast milk. The black dots again refer to the observed breast milk levels of sucralose. The green lines represent the predicted concentration, and they're using a transit compartment, like a delayed compartment into the breast milk, did help with adequately describing the breast milk profiles. And this is the overall fit in breast milk. Here, the plasma to breast milk coefficient, partition coefficient was about 0.18 or 18%, which corroborated with the observed data. And the postpartum age was able to explain some variability in this partition coefficient, which probably mechanistically we can think of because of changing in the milk constitution over a period of time. A postpartum age could be a potential covariate that could explain the partition coefficient of sucralose. So using the the plasma and the breast milk model, then we kind of try to understand that was from a single administration. So if mother consumes, let us say, in a typical alternate, I mean, in a real life scenario where mother could consume up to 3 beverages or 3 times in a day, 1 beverage 3 times in a day. So that this is how the maternal plasma looks like. As you can see, it is accumulating as more number of consumption with increased consumption, and this is how the breast milk looks like. And subsequently, the infant levels for a typical average infant about 13 weeks, assuming a weight of 6 kgs and 8 feeds per day. When the infant consume milk at 1 hour, 5 hour, 7 hour. This is a general scenario we want to test and see. So you could see the infant levels. Obviously, the more the mother consumes, you can see a higher level in the sucralose plasma concentration. Just make sure, I mean, please pay attention that the scales are very different. So obviously, the infant levels are much lower. About 0.09 nanogram per ml, around the Cmax of about 0.1 nanogram per ml compared to the breast milk levels, which is about 15 nanograms per ml. So if we really calculated the RID, the relative infant dose, it was less than 0.05% for sucralose. But having said that, we are still looking into what could be the clinical implication of these repeated consumption of LCS containing beverages, and what does this mean for infant? Meaning sucralose is known to have even with lower quantities, I mean, sweetness index is much more higher. So we don't know exactly what implications would it have on infants. So those are some things which we are still working on and trying to understand. But at least this exercise helped us to have an idea about what are the levels that we are expecting to see in a real life consumption of these low calorie sweeteners. So those are our next steps in terms of making some observations about the levels that we are seeing in infant. How do we clinically explain them? Is it clinically relevant? And maybe potentially inform some breastfeeding guidelines for LCS consumption. So this concludes the first case study. The second case study is predicting infant exposure to THC via breast milk in recreational cannabis users. So here, the question was, we leveraged a PBPK modeling approach because it is such a study cannot be obviously conducted, or it has a lot of logistical challenges to ask a breastfeeding, to do a control study, in other words, meaning asking a breastfeeding woman to take cannabis, or a woman taking cannabis after to breastfeed a baby, and then collecting levels is not logistically possible. So in that case, we leveraged a PBPK modeling approach, and this is by my student Jide. So some background about prevalence of cannabis use in lactating women. There are potentially health benefits for medical cannabis use in pain relief, appetite stimulation. There are also self-reported benefits of recreational cannabis users. Working with one of the clinical collaborator who works in substance use disorders, as per her, at least 90% of the women that come to the clinic are like recreational cannabis users for whom a decision has to be made on whether the woman can breastfeed or not. And it's a scary statistics here as well. As per CDC, in US, there are about 48 million cannabis users, greater than 12 years old, and among them, 43% of women, and among them, there are majority of them are in the 18 to 45 years age range, and there is a self-reported increase of self-reported cannabis use during pregnancy and breastfeeding, which with it is no surprise as more and more states become legalization of cannabis. This is known to increase. So the question here, actually, this was a question that the clinician was asking us. So I'm like a practicing OBGYN, talking with a patient who is a recreational cannabis user, and what kind of a decision should I make? Should I tell her to breastfeed or not? Although there are guidelines in ACOG, there are a lot of uncertainties, and for at least 55% of the in one of the publications, they said 55% actually tell them not to breastfeed. But knowing the benefits of breastfeeding, this was a very practical question that we wanted to address. So should mothers who use cannabis breastfeed or not? That's a very practical question. In other words, we wanted to know what is the PK profile of THC, which is the psychoactive component of cannabis, tetrahydrocannabinol, and how much gets into the infant. There are quite a few studies, observational studies, which have studied THC in breast milk. It is known to get into breast milk. It's a milk to plasma ratio ranging from 2 to 35, which is a wide range. And in this case, the use of a PBPK modeling approach was more appropriate. So I think Dr. Fletcher talked about this approach. Our general workflow was starting with an IV PBPK model, and then in adults, and then feminized the parameters, and then we studied an oral inhalation model because majority of the recreational cannabis users smoke joints. So that's why we use that as the predominant route of administration. And then we had an oral inhalation model, and then we incorporated the breast milk model into it, a lactation model. And then for the infants, there was no data, but we used one of the adult trial and then scaled it down to infants for us to verify this model. So this is the model structure. As I said, this was the oral inhalation component, where we considered 4 different compartments in the lung to understand the deposition of cannabis or THC in these compartments. And then we have the breast milk maternal compartment here. This is the lactation PBPK model, and then which was used to which was linked with the infant PBPK model for the dose, from the dose. So again, as I said, smoking is the predominant route of recreational cannabis use. And these are a couple of model qualification plots for the PBPK model. And this was for the intravenous THC. There were several studies which had intravenous THC in adults. So the blue trials are the one which we used for training the PBPK model, and the pink ones are the one which we use for model qualification or model validation. This is the AUC ratio and the Cmax ratio, and they are within the pretty much within the 2-fold range. And this is for the smoked THC. We had several studies based on which the black studies are the ones which was used to train the model, and the pink studies are the one which we used to validate. And you can see that the PBPK model was adequately qualified in both intravenous and describing the intravenous and smoking in adults for THC. Then we wanted to see how the breast milk model is performing, or the lactation PBPK model. And there were 2 studies from which we obtained some data. One was a breast milk study, which had concentrations collected up till 4 hours. Then there was another study which had some concentration measured at 120 hours, or maybe roughly 5 days. But there were a lot of uncertainty here about, here we were pretty much clear on what dose they were taking in terms of THC. The other study did not have that information, so we had to guess a little bit. But these are the blue lines are the model predicted lines to show that we are able to reasonably, the breast milk model is able to reasonably explain the data that is observed in the literature. So that gave us confidence that, yes, we can use it to simulate conditions which could be a real life scenario so that the question can be answered. And this is also in the infants for different age groups. This is neonates to 1 month old, 1 to 3 months old, 3 to 6 months old, and 6 to 12 months old. And the pink line is the information that is scaled from an adult oral model to an infant, to the infant levels. And the blue ones are the PBPK model predicted. So we are reasonably doing a decent job here in terms of scaling it down. One thing I have to mention is THC is extreme, it's metabolized. The predominant enzymes are CYP2C9 and CYP3A4. So corresponding age maturation factors were considered for these lower infant age groups. So having the PBPK model adequately qualified, we then evaluated scenarios of what happens if the mother smokes 2 times or 3 times, perhaps not 6 times is not a realistic scenario, but we wanted to see in terms of how the concentrations look like. And certainly here, there's no surprise. This was as expected. The THC gets into the breast milk. So you can see the blue lines, which are the breast milk levels much higher. It's still accumulating as compared to the plasma levels. And essentially that would have an implication in terms of the infant dosing. So these are the infant levels. It's a busy graph, but what I would like to point out is the Cmax in all the different age groups that we studied up till 12 months, the Cmax ranged between 0.1 to 0.15 nanogram per ml up to 6 months old. So what does this level mean? Meaning, how do we interpret these levels? So there are some studies that is done in adults, like THC levels greater than 2 nanogram per ml have reported some kind of cognitive impairment. Driving impairment may be relevant for infants, but that was some kind of a target level that showed some kind of impairment in adults. But there is also another aspect in terms of safety, because these levels have also been shown to increase heart rate in adults. So in terms of infants, although we may be considering these levels are much lower, 13 times lower as compared to the levels that is seen in adults, but the immediate in terms of a safety aspect, there is an increased heart rate that has been observed. So some careful considerations needs to be done by the clinician when they have to make a decision. But having said that, this exercise at least helped us to have some kind of an information that's available that we can use for some objective decision making, that the clinicians can keep in their mind to make their decision about when they have to talk to a patient and say, okay, should I breastfeed or not, in terms of providing them recommendations. As I said, the levels in breastfed infants were 13 times lower in adults, but certainly there are in terms of safety aspects, in terms of adverse events, there is an increased heart rate that needs to be kept in mind. So, but this gives the project helped them, or the research at least informs the clinician so that they can make a better informed decision making when they are interacting with the patients. So these are the 2 projects that I thought would be where we used well leveraged pharmacometric models that can potentially inform answering more clinically relevant questions. So I would like to thank the George Washington group, which was part of the low-calorie sweeteners trial. Dr. Alison Silvetsky, she's the PA, and this is the entire group which worked on conducting the clinical trial and bioanalysis. And for the cannabis project, I would like to thank my collaborator, Dr. Cash, for providing his expertise on PBPK modeling, and Dr. Katrina Mark, she's the OBGYN who came up with the original question, as she regularly interacts with patients in the Baltimore area, so you can imagine. And this is my group at the Center for Translational Medicine. I would like to thank them, and I'll be happy to take any questions.
Thank you very much for your great talk. I'm Yuki Otani from Eli Lilly and Company. So because we also don't have much of evidence or testing that we've done in these kind of breast milking studies, I want to ask you, I guess, a clarifying question, especially on the first study that you were talking on, the PK modeling on the small molecule. So I think the predictions are great. So I wanted to get an insight on one, what kind of like breast milk sample that you use, like how much volume, like at what type of points you were you taking like both breasts or just one or just a little bit of it or that kind of thing. And the second one is if you would also to have a study with, let's say, like a longer half-life work, like large molecules and things like that. Is there any kind of consideration that we should take for the sampling timing scheme for those studies?
Yeah, that's a great question. So in our study, this is a small molecule, and the samples were collected. It was exclusively breastfed women, so they did breastfeed. So it was mainly from typically from one of the breasts. That was what was considered. Whatever that the child could take, the infant could take it from. And in terms of but there are some women who actually used a breast pump. So we wanted to make sure we collected both fore milk and the hind milk because the composition of the milk is different. So I think we had a volume of about 15 ml of breast milk sample that was collected, so that it comprises of both the initial portion and the latter half of the breast milk collection. To your question about large molecules, that's a good question. I mean, they know you have a long half-life of about 2 weeks. So perhaps, I mean, I think you probably can design based on the plasma, how you would collect plasma and maybe stagger the breast milk samples in those cases could be a reasonable starting approach, I would think. Yes. Yes. Yes, that's correct.
I have an online question. Is there any preclinical data that could inform the infant safety threshold for THC?
Preclinical data?
Yeah.
There could be information. I'm pretty sure about that. We are still looking into it, but I think that is one aspect that we are, for us to make meaningful interpretations of the levels, we are looking into it. As of now, I don't recall having an answer for that. Yeah.
Hi, Wojciech Krzyzanski, University of Buffalo. I have a questions regarding handling the dose and the bioavailability, especially for this THC compound that is inhaled. So I understand, the basic measure, clinical one, is just puffs or number of smokes. But then, pharmacologically, how do you translate a smoke or a puff into actual dose that gets to the systemic circulation? So how do you do it from the modeling perspective? So it's like percentage of a THC in each puff, and then so that's my question.
Yeah, that's a great question. So for the oral inhalation model, like we considered 4 compartments, and this model was based on an International Commission of Radiological Products. This was more developed for smoking, like nicotine. So we adapted that model here where we had 4 compartments, and the fraction of smoke deposited in each region was calculated based on based on parameters that were available from the smoke. But your question about bioavailability, the smoking has a wide range of bioavailability. It's hugely variable because, as you can imagine, at least in the literature, I think it's about like 15 to 30% is the absolute bioavailability in terms of smoking as compared to an IV route. So I think this was when we tried to understand, when we calculated the fraction deposited in the 4 compartments, we kind of made sure that was in range with what you would probably see like in terms of absolute bioavailability, in terms of.
So you assume some bioavailability, I guess.
Yes.
And then did you include some variability of that bioavailability? Okay, thank you.
That concludes today. Thank you so much. That's a wonderful presentation.
Right, we're going to break for lunch now for the next